Welcome to the Deep Dive. Today we're venturing into, well, one of the body's most incredible yet often overlooked structures, the human ear.
It really is remarkable.
It's so much more than just the bit we see. We're going to uncover this hidden world behind both hearing and balance using histology, a text and atlas, 8th edition, as our guide.
And what's fascinating looking at it from a histological perspective is how the microscopic structure, the cells and tissues, directly explains how it works.
And why it sometimes doesn't work, I suppose.
Exactly. Understanding the blueprint helps understand the function, and yes, the common clinical issues too. It's all connected at that tiny level.
Okay, so let's start big picture. The ear's grand design. It's basically a three-part sensory organ, right? External, middle, and internal ear.
That's the fundamental layout.
And it pulls double duty hearing and balance. How does one small organ manage two such different, really complex jobs?
Well, that's the genius of it. Each of those three divisions, external, middle, internal, plays a part in both systems, though some parts are more specialized. Think of sound first. The external and middle ear act like collectors and conductors. They gather sound energy.
Funnel it in?
Precisely. Funnel it towards the internal ear. That's where the specialized sensory receptors take over.
And they do the conversion from sound waves to what?
To electrical impulses, the language the brain understands, but at the same time, the internal ear houses completely separate vestibular receptors.
For balance.
For balance. They're constantly monitoring gravity, head movements.
Keeping us upright, coordinating our eyes. It's amazing.
It really is.
And developmentally, where does all this intricate stuff come from? I read it starts incredibly simply.
Almost unbelievably so. It begins with the surface ectoderm, just a thickening called the otic placode on the side of the developing head.
Just a patch of cells.
Initially, yes. It then folds inward, forming the otic vesicle, which is the precursor to the entire membranous labyrinth, the delicate inner sacs and tubes, and other parts arise nearby. The pharyngeal arches, for example, contribute to the auditory tube, the middle ear lining, even the ossicles, those tiny bones.
So the malleus and incus from the first arch, stapes from the second. It's all mapped out early on.
Exactly. And this shared origin explains the tight connections, like with cranial nerve 8, and how everything integrates. You can almost visualize it if you think about figure 25.1 in the text, showing those three main divisions.
Okay, let's zoom in on the first division then, the external ear. The visible part, the auricle or pinna. It's got that characteristic shape because of elastic cartilage, right?
Correct. Covered by thin skin, which has hair follicles, sweat glands, sebaceous glands, the usual skin components.
And its job is mainly sound localization, helping us tell where a sound is coming from, though maybe less crucial for us than for some animals.
It contributes, yes, and provides some minor amplification. It's often described as nearly vestigial in humans compared to, say, a bat or a dog that can swivel its ears dramatically.
Then the sound goes down the tube, the external acoustic meatus.
Right, the ear canal, an air-filled S-shaped tunnel about 2.5 centimeters long, leading right to the eardrum.
And it's part cartilage, part bone?
The outer third is cartilaginous, the inner two-thirds are bony, within the temporal bone, and importantly, it's all lined by skin.
And this skin, especially in that outer part, has special glands. You mentioned hair follicles and sebaceous glands, but also ceruminous glands.
Yes, specialized coiled tubular glands. Their secretions, combined with the sebum from sebaceous glands and desquamated epithelial cells, shed skin cells.
Makes earwax, cerumen.
Precisely. And cerumen is fascinating from a histological and functional standpoint. Because that ear canal is a blind pouch, dead skin cells can't just fall out.
Ah, so the wax carries them out.
It's a self-cleaning mechanism. The wax, along with trapped particles and shed cells, slowly migrates outwards. It also lubricates the skin, prevents dryness, traps dust, and insects. And it provides antimicrobial protection. It's quite a clever substance.
But sometimes it gets blocked. You mentioned clinical correlations. Impacted cerumen.
Exactly. If it builds up too much, it can completely block the canal.
And since sound needs to travel through there.
It causes hearing loss, conductive hearing loss, because the conduction of sound is blocked.
That's the one. A common, usually easily fixed cause of hearing difficulty.
Okay, moving inwards. Past the eardrum lies the middle ear, an air-filled space.
Yes, the tympanic cavity. It's housed within the temporal bone. And inside, you find those famous auditory ossicles, the three smallest bones in the body.
Malleus, recus, stapes.
Along with the auditory tube connection and tiny muscles attached to the ossicles.
Sounds like a busy little room. What's its main function?
It's essentially a transformer. It takes the vibrations arriving through the air in the ear canal.
Hits the eardrum.
Right. And it converts those air vibrations into mechanical vibrations of the ossicles. These vibrations are then transmitted efficiently to the fluid-filled internal ear.
Via specific points, right? The oval and round windows.
Exactly. Those are the interfaces with the inner ear. The middle ear overcomes the impedance mismatch between air and fluid. Impedance mismatch, meaning it's harder to vibrate fluid than air.
Much harder. The middle ear system amplifies the pressure to make it happen.
And the key player initiating this is the tympanic membrane, the eardrum. It's not flat, is it?
No, it's cone-shaped, pointing slightly inwards, about a centimeter across, and it has 3 distinct layers, histologically speaking.
Skin on the outside?
Continuous with the ear canal skin. Then a core of connective tissue, mainly collagen fibers, arranged radially and circularly, this gives it strength. The pars tensa, or tense part.
And inside.
A mucous membrane, continuous with the lining of the middle ear cavity. There's also a small, looser area at the top, the pars flaccida, lacking that fibrous layer.
And you can see its shape during an exam, that cone of light.
Yes, the concavity reflects the otoscope light in a characteristic way. Its vibration is obviously critical. And the malleus, the hammer bone, is the only ossicle directly attached to it.
So the malleus vibrates, then the incus, then the stapes.
Correct. They form a lever system. Malleus, hammer, incisticus, anvil, stapes, stirrup. This lever action plus the size difference between the large eardrum and the small footprint of the stapes on the oval window.
That's the amplification mechanism.
That's the pressure amplification. It ensures the energy gets transferred effectively into the inner ear fluid.
Are there muscles involved too?
2 tiny ones. The tensor tympani, attached to the malleus, increases tension on the eardrum, and the stapedius, attached to the stapes, dampens its movement at the oval window.
Why would you want to dampen the movement?
Protection. It's called the attenuation reflex. In response to loud sounds, these muscles contract, stiffening the ossicular chain and reducing the vibration transmission.
Ah, like built-in ear protection.
A rapid involuntary one. It helps protect the delicate inner ear structures.
Then there's the auditory tube, or eustachian tube. We mentioned it connects to the middle ear.
Right. It runs from the anterior wall of the middle ear cavity down to the nasopharynx, the back of your throat.
And it's usually closed.
Normally flattened and closed. But it opens during swallowing, yawning, or chewing.
To let air in or out. That's the popping sensation.
Exactly. It equalizes the pressure in the middle ear with the outside atmospheric pressure, essential for the eardrum to vibrate properly. Histologically, it's lined mostly with ciliated, pseudostratified columnar epithelium, like much of the respiratory tract.
But this connection is also a route for infection, isn't it?
It is. Infections from the throat can travel up the tube into the middle ear. That's otitis media, a common childhood illness.
And you mentioned mastoid air cells earlier.
Yes, these are air-filled spaces in the mastoid process, the bony bump behind your ear. They connect to the middle ear cavity and share the same mucous membrane lining.
So infection can spread there too, mastoiditis.
It can, and it used to be quite dangerous before antibiotics. It's less common now, but still a potential complication.
Okay, another clinical point related to the middle ear, otosclerosis. You mentioned bone overgrowth.
Yes, it's an abnormal bone remodeling process in the temporal bones, specifically around the ossicles.
And it particularly affects the stapes.
Often, yes. The bone growth can fix the stape's footplate into the oval window, preventing it from vibrating.
Which stops the sound transmission.
Exactly. It causes progressive conductive hearing loss. The sound just can't get into the inner ear efficiently.
Is the cause known?
Not entirely. There's a genetic component, and some association with measles virus has been suggested, but it's complex. Treatments range from hearing aids to surgery.
Like replacing the stapes.
Well, stapedectomy, yes. Removing the fixed stapes and replacing it with a prosthesis can sometimes restore hearing remarkably well.
Fascinating. Okay, let's finally venture into the deepest part, the internal ear. This is where the real sensory action happens for both hearing and balance.
Indeed. It's housed deep within the petrous part of the temporal bone, the densest bone in the body.
And it has this complex structure labyrinth.
2 of them nested. There's the bony labyrinth, which is a system of cavities and canals carved into the bone. And suspended within that is the membranous labyrinth, a delicate system of interconnected sacs and ducts.
So bone outside, membrane inside. What's in the spaces?
Fluid. And crucially, different fluids in different spaces. This is key.
Right.
Inside the membranous labyrinth, you have endolymph. It's unique, very high in potassium ions, low in sodium, almost like intracellular fluid.
And outside the membrane, but inside the bone.
That space, the paralymphatic space, contains paralymph. It's much more like extracellular fluid or cerebrospinal fluid, low potassium, high sodium.
So two different fluid environments separated by the membrane.
Yes. And there's actually a third tiny space within the organ of Corti itself containing cordylymph, which is similar to paralymph. These distinct ionic environments are absolutely critical for generating the electrical signals in the sensory cells.
Where do these fluids come from?
Endolymph is actively secreted by a specialized tissue in the cochlear duct called the stratia vascularis. Paralymph is thought to be partly an ultrafiltrate of blood plasma and partly derived from cerebrospinal fluid. They have separate drainage routes, too.
This sounds incredibly finely balanced.
It is. Maintaining these fluid compositions and pressures is vital.
Now, the bony labyrinth itself, what are its main parts? You mentioned canals.
Right. Looking at figure 25.7 helps visualize this. There are three main interconnected spaces. The semicircular canals for balance, the central chamber called the vestibule, also balance, and the snail-shaped cochlea for hearing.
And the oval and round windows connect here.
The oval window, where the stapes fits, opens into the vestibule, connecting the middle ear to the paralymphatic space. The round window is a pressure relief valve at the base of the cochlea, also covered by a membrane.
And the membranous labyrinth fits inside these bony shapes.
Perfectly. You have the semicircular ducts within the canals, the utricle and saccule, two sacs within the vestibule, and the cochlear duct spiraling within the cochlea, all filled with endolymph, all suspended in paralymph, referencing figure 25.8c.
Okay, now we're getting to the core sensory parts. Where are the actual receptor cells located?
They're clustered in 6 specific regions within that membranous labyrinth. Figure 25.8d shows these.
6 regions.
Yes. 3 cristae ampullaires, one in each semicircular duct. These detect angular acceleration, head rotation.
Right.
2 maculae, one in the utricle and one in the saccule, located in the vestibule. These sense gravity and linear acceleration, head position, moving forward, backward.
And the last one.
The spiral organ of Corti, located within the cochlear duct. This is the sound receptor.
And all these regions contain similar types of sensory cells.
They all rely on the same fundamental cell type, the hair cell. These are specialized epithelial mechanoreceptors.
Mechanoreceptors, they sense physical movement.
Exactly. They are the mechano-electrical transducers. They convert mechanical force, whether from sound waves or head movement, into an electrical signal.
And they're called hair cells because?
Because of the bundle of projections on their apical surface facing the endolymph. This isn't true hair, but a bundle of stereocilia.
Stereocilia, like stiff little hairs.
Very stiff, yes. They're arranged in rows of increasing height, like a staircase. In the vestibular system, there's also one true cilium, the kinocilium, at the tall edge, which defines the cell's polarity. See figure 25.9.
Polarity, meaning direction matters.
Absolutely. The direction the stereocilia bend is critical for signaling.
How are these stereocilia structured? What makes them stiff and able to signal?
Internally, they have a core of tightly packed actin filaments cross-linked by proteins like fibrin and aspin. Aspin mutations actually can cause deafness and balance issues because the stereocilia aren't rigid enough. See figure 25.10.
Okay, so they're rigid. How does bending them cause a signal?
This is the amazing part. Tiny protein filaments called tip links connect the tip of each shorter stereocilium to the side of the adjacent taller one.
Like little ropes connecting them.
Exactly. And these tip links are directly connected to mechanically gated ion channels or meat channels located near the tips. Think of the tip link pulling the channel's gate open or letting it close.
So bending towards the tallest edge.
Stretches the tip links, pulling the meat channels open. Since endolymph is rich in potassium, K+.
Potassium rushes into the cell.
Right. K+ influx depolarizes the hair cell. This opens voltage-gated calcium channels further down, causing neurotransmitter release at the base.
And bending the other way.
Bending away from the tallest edge relaxes the tension on the tip links, allowing the meat channels to close. K+ influx stops, and the cell hyperpolarizes, reducing neurotransmitter release.
It's like an incredibly sensitive directional switch.
Extremely sensitive and incredibly fast.
How does the cell release neurotransmitters so quickly and reliably?
Using specialized synapses called ribbon synapses. figure 25.11. Instead of vesicles just floating randomly, these synapses have an electron-dense structure, the ribbon, which tethers lots of vesicles right near the release site.
Like having ammunition ready to fire.
Exactly. It acts like a conveyor belt, allowing for rapid, sustained release of neurotransmitters, mostly glutamate, for precise temporal signaling.
You mentioned type I and type 2 hair cells earlier in the vestibular system.
Yes. Figure 25.10. Type I cells are more flask-shaped and are almost completely enveloped by a single large afferent nerve ending called a chalice. Type 2 cells are more cylindrical and have multiple smaller Bouton-type nerve endings. They likely process information slightly differently.
Okay, let's apply this. How does the crista ampullaris work in the semicircular canals? Figure 25.12.
The crista is a ridge covered with hair cells and supporting cells. Their stereocilia project into a gelatinous, dome-shaped structure called the cupula, which spans the duct.
So the cupula blocks the canal.
It acts like a swinging door. When you rotate your head, the bony canal and the cupula move, but the endolymph inside lags behind due to inertia.
The fluid pushes the cupula.
Yes, the lagging endolymph pushes the cupula, bending the stereocilia of the hair cells embedded in it. This signals angular acceleration to the brain.
Got it. Now, the maculae, the utricle and saccule, figure 25.42, they sense linear movement and gravity.
The maculae are patches of hair cells and supporting cells, again facing the endolymph. But here, the stereocilia project into a different structure, the otolithic membrane.
Otolithic, like ear stones.
Exactly. This gelatinous membrane contains tiny crystals of calcium carbonate called otoliths, or otoconia. These make the membrane heavy, denser than the endolymph.
So gravity affects it.
Yes. When you tilt your head, gravity pulls on the heavy otolithic membrane, causing it to shift and shear across the hair cells, bending the stereocilia.
And linear acceleration, like in a car.
Same principle.
Yeah.
When the car accelerates forward, the otolithic membrane lags behind slightly due to inertia, again shearing across the hair cells. This signals linear motion.
So elegant. Okay, finally, the main event for hearing. The spiral organ of Corti, figure 25.9. It's inside the cochlear duct, right?
Yes, sitting on the basilar membrane within the cochlear duct, also called the scala media. Remember, the cochlear duct divides the bony cochlea into 3 parallel chambers, or scalae.
Scala vestibuli above, scala tympani below, both with paralymph.
Correct. And the scala media in the middle, filled with endolymph containing the organ of Corti. These scalae spiral up the cochlea and connect only at the very apex via a small opening called the helicotrema.
And the walls of this scala media are important.
Very. The roof is the thin vestibular membrane, or Reisner's membrane. The outer wall is the stereocilia vascularis, that crucial tissue that produces endolymph and maintains its ionic balance. It's highly vascularized and metabolically active.
And the floor.
The basilar membrane. This is fundamentally important. It supports the organ of Corti, and it varies in width and stiffness along the length of the cochlea. Narrow and stiff at the base, wide and flexible at the apex.
That variation is key for pitch perception, isn't it?
Absolutely. We'll get to that. Now, the organ of Corti itself is complex. See figures 25.21, 25.22. It has the sensory hair cells' inner and outer rows, but also several types of supporting cells.
Like phalangeal cells and pillar cells.
Yes. Pillar cells form the rigid triangular tunnel of Corti in the center. Phalangeal cells support the hair cells, both physically and metabolically. Crucially, the apical surfaces of these supporting cells and the hair cells join together via tight junctions to form the reticular lamina.
A barrier.
A critical barrier. It separates the K+ rich endolymph in the scala media above from the cordolymph, similar to paralymph, bathing the lower parts of the hair cells below. This separation is vital for the electrical currents during transduction.
And there's one more membrane, the tectorial membrane.
Yes. This gelatinous ribbon extends from a structure called the spiral limbus and lies over the organ of Corti.
Do the hair cells' stereocilia touch it?
The tips of the stereocilia of the outer hair cells are embedded in its underside. The inner hair cell stereocilia might make slight contact or just be moved by fluid coupling. This interaction is essential for sound detection.
Okay, put it all together. How do we actually hear sound? Figure 25.23.
All right, the journey. Sound waves hit the eardrum. Vibration travels through malleus, incordus, stapes. Stapes pushes the oval window.
Creates pressure waves in the paralymph.
Starting in the scala vestibuli. These pressure waves travel up the cochlea. Because the scala media's walls are flexible, the pressure wave passes across the vestibular membrane into the endolymph and then displaces the basilar membrane downwards.
pushing paralymph in the scala tympani towards the round window, which bulges out to release the pressure.
Exactly. The key is the displacement of the basilar membrane. This movement isn't uniform.
Because of its varying stiffness.
Precisely. The pressure wave creates a traveling wave along the basilar membrane. High-frequency sounds cause the base of the membrane, narrow, stiff, to vibrate maximally. Low-frequency sounds create maximum vibration near the apex, wide, flexible.
So the location of maximum vibration tells the brain the pitch.
That's the place principle of frequency discrimination, and the amplitude of the vibration at that location determines the perceived loudness.
But how does the vibration translate to hair cell stimulation? You mentioned the tectorial membrane.
This is the critical sharing effect, because the basilar membrane, where the hair cells sit, and the tectorial membrane, where their stereocilia are attached, are hinged at different points.
When the basilar membrane moves up and down,
It creates a shearing or sliding motion between the two membranes. This bends the stereocilia back and forth.
bending the stereocilia opens and closes those meaty channels.
Bingo! That generates the electrical signal in the hair cells, neurotransmitter release, and firing of the cochlear nerve fibers connected to them. The brain interprets this pattern of nerve firing as sound.
Incredible mechanism. And the nerve carrying this information is the cochlear nerve.
Yes, part of the vestibulocochlear nerve, cranial nerve 8th. It branches. The vestibular nerve collects balance info from the cristae and maculae, cell bodies in the vestibular ganglion.
And the cochlear nerve collects hearing info from the organ of Corti, cell bodies in the spiral ganglion within the cochlea itself.
Correct.
Where did these signals go in the brain?
Both nerves enter the brainstem. Vestibular signals go to vestibular nuclei, then influence posture, balance via connections to the cerebellum, spinal cord, and eye movement centers. Cochlear signals travel through several auditory nuclei in the brainstem and midbrain.
Relays.
Yes, processing stages. Then to the thalamus, and finally project to the primary auditory cortex in the temporal lobe, where sound is consciously perceived.
And the brain can talk back. Efferent fibers.
Yes, there are descending pathways from the brainstem back to the hair cells, particularly the outer hair cells. These can modulate the sensitivity of the cochlea, helping us focus on sounds or protect against noise damage.
And the blood supply is crucial, you say.
Extremely. The labyrinthine artery, usually a branch of a cerebellar artery, is the sole supply to the membranous labyrinth. It's an end artery, no backup collateral circulation.
So a blockage is catastrophic.
It can cause sudden, complete hearing loss and vertigo on that side. It highlights the delicate nature of this system.
Which brings us back to clinical issues. We touched on conductive hearing loss. What about sensorineural?
Sensorineural hearing loss involves damage to the inner ear itself, the hair cells, the cochlear nerve, or central auditory pathways.
Causes.
Many things. Prolonged exposure to loud noise, acoustic trauma, certain viral infections like mumps or measles, some medications, ototoxic drugs, genetic factors, the natural aging process, presbycusis, Meniere's disease.
And this affects sound clarity, not just volume.
Often, yes. Hair cell damage can distort sound perception. Cochlear implants can help some individuals by bypassing damaged hair cells and directly stimulating the cochlear nerve endings electrically.
And balance disorders, vestibular dysfunction.
That often presents as vertigo, a true sensation of spinning or movement when there is none, or general dizziness, unsteadiness.
What causes that?
Again, various things. Viral labyrinthitis, infection of the inner ear, benign tumors like acoustic neuromas pressing on the nerve, certain drugs.
And you mentioned BPPV before the crystals.
Benign paroxysmal positional vertigo. Those little otoconia crystals detach from the macula, usually in the utricle, and float into one of the semicircular canals, most often the posterior one.
So they move when they shouldn't.
Exactly. When the person moves their head into certain positions, the loose otoconia move within the canal, inappropriately stimulating the cupula and causing a brief but intense spinning sensation, usually treatable with specific head maneuvers.
And Meniere's disease. Fluid pressure.
Yes, Meniere's is thought to involve excessive accumulation of endolymph within the membranous labyrinth endolymphatic hydrops. This increased pressure disrupts both hearing and balance functions.
Leading to?
Episodes of vertigo, fluctuating low-frequency hearing loss, tinnitus, ringing in the ear, and a feeling of fullness in the ear. The exact cause of the endolymph buildup isn't always clear.
It really underscores how interconnected everything is in that tiny space. Hearing, balance, fluid pressures, microscopic cells.
It's an absolutely exquisite piece of biological machinery. The precision required at the cellular and molecular level to give us our sense of sound and spatial orientation is just staggering.
From collecting sound waves to decoding complex harmonies and keeping us balanced, all within a structure tucked away inside our skull. An incredible symphony of mechanics and biology.
What's truly astonishing, I think, is how finely tuned these microscopic structures really are. Their precise function underpins our very experience of the world, our connection to it through sound, our ability to navigate it through balance. This raises, for me, an important question.
Okay.
How does our growing understanding of these incredibly delicate systems, right down to the molecules in a tip link or the proteins in a rib and synapse, how does this knowledge inspire new ways to protect these vital senses? Or maybe even, one day, fully restore them when they're lost?
That's a powerful thought to end on. How can we use this intricate knowledge for tangible benefit? Something for all of us to consider. Thank you for joining us on this deep dive into the microscopic marvel that is the human ear. Keep exploring the body's hidden complexities.